my pleasure to introduce the second speaker in this session, Dr. Shimabukuro from CDC. He's been at the vaccine safety component of CDC since 2010 and has led many of the studies on post-licensure or post-EUA approval of COVID vaccines in the USA. So he's going to give us the benefit of his take on real-world evidence in relation to decision-making and presumably how it fed into the EUA approval process.
So as the cover slide says, I'm with the Immunization Safety Office at CDC, and we do phase 4 safety assessments, so post-authorization and post-licensure. So I'm going to be more on the public health decision-making and leave most of the EUA and licensure stuff for my colleague Hector. Just brief agenda. I'm gonna do some background, then move into 2 specific case examples that I think you're all familiar with, so I'm not breaking any new ground here, but I think they're instructive on how these case examples informed CDC decisions, and then wrap it up with conclusions. So, just for background, we use multiple systems for signal detection, signal assessment, and clinical consultation. Our passive spontaneous reporting system is the vaccine adverse event reporting system that's co-managed by CDC and FDA. And for COVID, we are fortunate to have our passive surveillance supplemented by doses administered data from the CDC COVID data tracker. Our active surveillance system is the Vaccine Safety Data Link, which is an electronic health record-based surveillance and research system. We have our clinical consultation and technical expertise program, and then for COVID, we had a pregnancy registry, and we also had a new system for active surveillance called vSafe for COVID. So the first case example I'll do is the mRNA vaccines and myocarditis. So fairly early on, when the vaccine started being administered to younger individuals and children, we noticed increased reporting to our vaccine adverse event reporting system. And as I said, fortunately, during the pandemic, we had denominator data. We had timely and accurate denominator data, which was key to our assessment of this adverse event. And this is a screenshot from a paper, and the bottom line on this paper was based on passive surveillance reporting in the US, the risk of myocarditis after receiving mRNA-based COVID-19 vaccines was increased across multiple age and sex strata and was highest after the second dose in adolescent males and young men. So this is a table from that paper, and on the left-hand side, you have age and sex strata with reporting rates to VAERS. And on the far right-hand side, you see expected reporting, expected counts or cases based on background rates. And I've highlighted what I think is the most striking finding, and that's in the 16 and 17-year-old male group. You see, after second dose, a reporting rate of about 105 per million, where expected would be about 1.3. And so this is clearly an imbalance and an evidence of an increased risk. Also in that analysis, when you plot out the onset of symptoms for these cases, we see that for both vaccines, Pfizer and Moderna, they tend to cluster shortly after vaccination, usually within a few days after vaccination. This data, I will say, is limited by, this is limited by reporting biases and the other limitations of passive surveillance, but I'll show you some information from VSD that basically confirms this. So this is a table from our Vaccine Safety Data Link system, and I think I should have probably provided a temporal scan statistics which showed the clear clustering in the first few days. But what this shows is basically risks in the 0 to 21 on the top and then 0 to 7 on the bottom. And you can see the adjusted rate ratio, which is a measure of relative risk, highly elevated in the 0 to 7 days, more so than in the 0 to 21 days just above it. But you'll notice on the far right-hand side, when you look at excess cases, which is a measure of attributable risk, you see that they look pretty similar, whether in the 0 to 21 or 0 to 7 days. And so what that really does is reinforce that within this risk interval that we looked at, 0 to 21 days, the risk is really concentrated in the 0 to 7 days. This is additional VSD data once we had enough data to look at product-specific estimates. So based on this information, FDA updated the regulatory language in the EUA documents and in the package inserts. This is language from the package inserts, but it was similar in the EUA documents, and that's post-marketing data demonstrated increased risk of myocarditis and pericarditis, particularly within 7 days following the second dose. So that looked at really the acute outcome of myocarditis, but we're also interested in the longer term outcomes. So CDC did a study looking at outcomes 90 plus days after the onset of myocarditis. And what we determined was that most individuals were judged by their healthcare provider, this is 80+%, as fully recovered or probably fully recovered. And we have some additional work ongoing where we're looking at that remainder that weren't, and most of those have also been judged as fully recovered or probably fully recovered. So these individuals tended to have favorable outcomes. So as a result of that, CDC updated its interim clinical considerations, which is really the implementation guidance for the ACIP and CDC recommendations. And you see here some of the current language: development of myocarditis or pericarditis after a dose of any COVID-19 vaccine is a precaution to a subsequent dose of any COVID-19 vaccine, and subsequent doses should generally be avoided. So now I'll move on to the second example, which is Janssen COVID-19 vaccine and TTS. So I think most of you are familiar with this condition. It was initially recognized with the AstraZeneca vaccine that was widely used in Europe. It's thrombosis, often in unusual locations, in the presence of low platelets. The onset observed with the Janssen vaccine was usually within 2 weeks of vaccination, and it had similar clinical features to heparin-induced thrombocytopenia. So this imbalance or this unusual reporting to VAERS was observed within weeks of the Janssen vaccine being introduced in the United States, and rapidly recognized. We were looking for it because we were aware of the AstraZeneca safety signal. But CDC released a health alert on April 13th and presented data to the CDC. And FDA and CDC recommended a pause in the use of the Janssen vaccine for several days for safety assessments. That was really to allow us to get a better handle on the data, to also put out the word to healthcare providers to be on the lookout for this condition and to get a vaccination history, and very importantly, to put out some clinical guidance on treatment, specifically that heparin should not be used, and they should use other types of anticoagulants because in this case, heparin could actually make the condition worse. So the ACIP assessment at that time was the benefits of resuming the Janssen vaccination among persons age 18 and older outweighed the risk and reaffirmed the interim recommendation under FDA EUA, which includes a new warning for the rare clotting events among women 18 to 49 years. So this was not the end of this assessment. More data were coming in, and we are continuing to evaluate this situation. So this is from a paper which included additional data showing the reporting rates in VAERS. And again, this emphasizes the importance of having denominator data, which we normally don't have in the United States. Looking at reporting rates for the Janssen vaccine compared to, on the very bottom there in red, you see the mRNA vaccines. And you see the reporting rate for the mRNA vaccines is just minuscule. It's 0.008 per million doses. And then you see the reporting rates for the Janssen vaccine tend to be elevated in these younger age groups, particularly in women of reproductive age, with a high of 10.6 per million doses in the 30 to 39-year-old female age sex strata. So this first major bullet here covers what I just showed you on the table, but also importantly, deaths from TTS after Janssen vaccine were estimated at about 2 per million doses in women aged 30 to 49 years. So really the ultimate vaccine safety outcome, clearly concerning there. And when ACIP reviewed this additional information in late December, they issued a statement. After reviewing the updated vaccine effectiveness and safety data, ACIP made a preferential recommendation for the use of the mRNA vaccines over Janssen in all persons age 18 and older in the United States. In May of 2022, FDA basically limited the authorized use of Janssen vaccine to individuals for whom other authorized or approved vaccines were not available, or for individuals who just refused to get an mRNA vaccine and would only get a non-mRNA vaccine. And then in June of this year, FDA revoked the EUA, and that vaccine is not going to be available moving forward in the fall. So just to sum up, there was a substantial amount of vaccination in the United States, 676 million doses, over 80% of the population getting at least one dose. So a lot of real-world evidence. CDC used established and new vaccines to monitor the safety of these vaccines. Timely and accurate doses administered or denominator information substantially enhanced the value of passive surveillance data from VAERS. Again, something we normally don't have for routine. That was unique to the COVID pandemic. The rapid assessment interpretation of COVID vaccine safety data during the pandemic informed vaccination policy and clinical considerations in near real time. And this real-world evidence contributed substantially to public health decision-making in the United States during the COVID vaccination program. And all these actions informed healthcare providers and policymakers and then protected and informed the public. So that concludes my presentation. I'll be happy to take questions.
Thanks very much, Tom. We've got any questions online, Peter? No, okay, we go straight to the questions from the floor.
Bob Chen, Brighton
from
collaboration again. Tom, very nice. So usually we kind of minimize the value of VAERS passive data, in part because it's kind of less than 1/4 of the 2x2 table, but in this outbreak it really kind of shined in some ways. Have you been able to kind of quantitatively compare, let's say, the reporting rates per million to the final attributable risk data that you've had through VSD to see, degree of underreporting, et cetera? How off was the passive data compared to the final true data?
First, I just want to say, VAERS has its limitations and has been criticized by many, but VAERS tends to give the most data the quickest and is very informative, especially during COVID. We did do some comparisons with the reporting rates to the incidence rates observed in VSD, and early on, they were fairly comparable. I think as time went on, the reporting dropped a little bit in VAERS, which you might expect. That tends to happen with the Weber effect. But early on, the reporting rates and the incidence rates that we observed in VSD were fairly comparable, yes.
Rebecca Chandler from CEPI. I want to thank you for your presentation, because I think that you have made, I just want to add to what Bob has said, I think you've made a very excellent demonstration of the fact that spontaneous or passive reporting is a form of real-world evidence, and it is very often left off the lists of real-world data sources. And I think that, to me, coming from a history of spontaneous reporting, it is real-world data. It's patient-centric. They're the ones who usually told you. So I think that COVID-19, I agree with Bob, I think it really demonstrated the value of spontaneous reporting, and I hope that moving forward, we will see spontaneous reporting systems on those lists of sources of real-world data.
Yeah, very good point. And I would say another early example of that was in the narcolepsy with swine flu issues, where very, very clear signals from passive reporting in the Swedish registry were put down to biased reporting, and it took a little while for even august committees like GAVAX from WHO to take this seriously. And the first study that was done in Finland really found difficulty in publication because it wasn't biologically plausible. And the kind of passive reporting, I think, can flesh out those things that you would never think of looking for. But alert clinicians pick them up because they know it's not part of their normal clinical pattern. Sorry about that. But I think it is a very important point. Thanks, Rebecca.
I just want to say, though, for especially for TTS, like we say this is VAERS data, but a lot of these initial reports came directly to CDC from clinicians picking up the phone and calling to CDC, and then we are able to route those into VAERS and do the follow-up. But I agree with you, kind of that astute clinician seeing something that looks suspicious and deciding to alert public health authorities. There's a lot of value in that, especially for really rare outcomes like TTS.
So that's 3 votes for passive reporting out of 3. That's good. I have 2 questions now based on that. First question is, since the denominator data was so useful, is there a way to continue receiving that so that you can continue better surveillance in the future?
The jurisdictional reporting requirements, by jurisdictional I mean basically state health department reporting requirements, they were basically written into the CDC provider agreement. So if you wanted to participate in the program, you had to agree to send data to the state health department. The state health department had to agree to send data to CDC. It's a labor-intensive process. It requires a lot of work and a lot of effort by providers and state health departments. I think it was definitely worth it in COVID, but I mean, that is a limitation to doing that just in routine times.
So what about doses distributed by manufacturers?
Doses distributed, we have that information. I think the problem there is not all doses distributed get administered or administered in a timely way. And then that doses distributed data may not always be available in real time. In COVID, those feeds to the COVID data tracker came in weekly. Now, there are some data delays, but on a weekly basis, we could just churn through and generate these reporting rates.